Good afternoon, everyone. I am Vennila Kanchana Devi Marimuthu from Sastra Deemed University. Today, I will be presenting the title Meta-Analysis Transcriptome Datasets Reveals Key Immune Gene Profiles and Signaling Pathways in Bastaris. This study for Introduction. This study focuses on integrating RNA data from multiple bovine diseases datasets to identify consistent immune responses. Through meta-analysis, we aim to uncover genes and pathways commonly altered across distinct infection conditions, which may serve as potential biomarker for disease resistance. Dataset associated diseases. First, a total of 4 gene expression datasets were retrieved from the NCBI Gene Expression Omnibus. These datasets represent RNA sequence data from Bastaris samples under various experimental conditions. The datasets were identified using the keywords expression profiling by high-throughput sequencing and immune responses using the GO database search. First, we have GSE 45439 related to bovine tuberculosis caused by Mycobacterium bovis. Second, we have GSE 125964 and clinical, subclinical postpartum uterine endometriosis. And GSE 62048 and GSE 247921 is related to John's disease caused by MAP. Workflow for RNA sequence analysis. We began with raw sequencing reads cleaned with FastQC for quality check and removed duplicates with the FastUnic. Then Trimatic for trimming the low quality reads and used Illumina Clip for adapter removal. Then aligned them to the Bastaris genome using Bautito. Gene expression was quantified with future counts followed by differential analysis using DECCB2. Finally, we conducted meta-analysis using R tool called meta-RNA-seq and did pathway enrichment analysis using Shinigo version 0.2. This integrated workflow helped us stitch together consistent signals across diverse datasets. Differentially expressing. From here, we found 28 significant genes. The green circle indicates the down-regulated genes and the maroon circle indicates the upregulated genes. Here, the most upregulated gene was PBX4 and then most down-regulated genes for LAPK. From here, we found the key immune genes from the 28 significant genes. The first is RGS2. It involves in G protein signaling, and PPARG. It involves in metabolism regulation, ZPP1, innate immune signaling sensing, RCAN, it's a calcineurin inhibition, IL-1A, it's an inflammation, TLR10, anti-inflammatory regulation, TAMD4, phagocytosis regulation, SBAGS is a cell cycle regulation it's used. Signaling pathway. From the 28 significant genes, we identified the 4 signaling pathways. CGMK PKG signaling. It regulates intracellular calcium levels. Oxytocin signaling pathway modulates cellular responses. Necropsis pathway controls programmed cell death. And osteoclast differential analysis involves immune receptors. Among the 4 signaling pathways, calcium signaling emerges as a central regulator across all pathways. Then, conclusion. Meta-analysis identified 28 significantly differentially expressed genes, including 8 immune-related genes, IL-1A, RGS2, RCAN1, TLR, showing the consistent expression changes across infections in Bastaris. Practical applications include potential biomarkers for ALD detection. Future improvements require experimental validation through gene expression assays, functional genomics, and integration of multi-omics approaches. This integrative transcriptome meta-analysis approach successfully uncovered conserved immune signatures and regularity mechanism. Come and see our posters and acknowledgement. Thank you.
Thank you very much for your presentation and for your nice work. So now we'll switch to the virtual